Okay, we are back. I had an internet outage. Apologize. Okay, so let's show. There we go. Uh, okay, so before the big break, we had begun discussing the cell wall and how it's different in different types of bacteria. Okay, the cell wall is going to help with the shape of the bacteria, and it gives it a strong structural support to keep the bacteria from bursting or collapsing under osmotic pressure. Okay, now what does that mean? Well, remember that when a cell enters an environment where the concentration of water is greater outside the cell than inside the cell, then one of the things that can happen is that the water can run into the cell and the cell can burst, right? So this would be our cell. Okay, there's our nucleus, right? And if we're in pure water, right, that water can rush into the cell. That'll cause the cell to swell up like an inflated balloon, okay? And then it'll pop, okay? Same thing is true in an environment where there's very little water outside the cell, right? If the cell is in an environment like that, okay, then water will leave the cell faster than it goes in, okay? And then the cell will shrivel up and it'll die, okay? Well, that's a process called osmosis, right? Water goes where it's least concentrated, or another way to say it is that water chases solutes, all right? And when you've got these layers of protection around you, then it helps bacteria from exploding or from disintegrating when the osmotic conditions change, okay? There are drugs, there are antibiotics that go after the structural integrity of the cell wall. And when the wall is weak, then when the cell hits, say, an environment where the water is going to rush in, we call that a hypotonic environment, then the cell's going to swell up and die. Okay, so that's how some of these drugs actually work. So if we look at the structure of the cell wall, we can see here, right, what peptidoglycan is all about, right? So peptidoglycan is made up of special sugars, okay? We abbreviate those sugars NAG and NAM, okay? NAG and NAM, all right? And what do those stand for? N-acetyl, N-acetyl glucosamine and N-acetyl muramic acid, all right? You don't have to remember that. Just remember NAG and NAM, okay? And they're connected to each other. You can see here how, right? They're connected to each other by special sugar-sugar bonds called glycosidic bonds. And that forms a stable, there we go. That forms a stable wall in one direction, right? In that direction, okay? But then we cross-link these things with little stretches of protein, okay? And that's what these guys are, right? So you've got cross connection and then you've got vertical connection, okay? And that makes for a pretty strong cell wall. So there's a NAM, all right? There's another NAM, and the NAMs are connected by this stretch of protein, okay? And then the NAGs are going to be on either side. There'd be a NAG up here, okay? And there'd be a NAG over here, okay? So NAG, NAM, NAG, NAM, NAG, NAM, all the way over and over again, okay? All right. Now, if we look at a gram-negative cell wall, you get a little bit of a different picture, okay? You can see here that you've got an inner and an outer membrane, and you've got peptidoglycan. So let's listen.
Most bacterial cell walls contain peptidoglycan. This structure is essential for maintaining the structural integrity of the cell. Peptidoglycan is a structure that is unique to bacterial cells. This combination of essential and unique makes peptidoglycan an excellent target for antibiotics. The precursor subunit of peptidoglycan, UDPN acetylmuramic acid, is synthesized in the cytoplasm. A lactyl group is added to carbon number 3 of UDPN acetylglucosamine to form UDPN acetylmuramic acid. 5 amino acids are added sequentially to UDP-NAM to form a pentapeptide side chain. Special adding enzymes are used to add these amino acids so that tRNA and ribosomes are not involved. In the case of Escherichia coli, L-alanine is added first, then D-glutamic acid, then mesodiaminopomelic acid. Finally, the dipeptide, D-alanyl D-alanine, is added to form the pentapeptide side chain. The NAM pentapeptide is transferred from UDP to bactoprenol phosphate at the membrane surface. Then NAG is transferred from UDP-NAG to the bactoprenol NAM pentapeptide to form the final disaccharide pentapeptide precursor. The completed NAM-NAG precursor is transported across the membrane to its outer surface by the bactoprenol carrier lipid. The disaccharide pentapeptide is then transferred to the growing end of the peptidoglycan chain to lengthen it by 1 repeat unit. As the bactoprenol pyrophosphate returns to the inside of the cell membrane, 1 phosphate is removed by a pyrophosphatase, leaving a bactoprenol phosphate to accept another NAM pentapeptide. Finally, the peptide side chains are linked together by transpeptidation reactions, forming peptide bonds. In E. coli, the free amino group of diaminopomelic acid is linked to the carboxyl group of the subterminal D-alanine, releasing the terminal D-alanine. Other enzymes remove the terminal D-alanine from side chains that are not cross-linked.
So again, okay, do I care about all of those particulars with the bactoprenol and all that stuff? No, not really, okay? What I care about is that you understand that the cell wall, peptidoglycan, are NAG-NAM polymers, okay, that are connected by, and let's make the protein different color. Hold on a second. Okay. But you're cross-linked by these little stretches of peptide that are coming off the NAM, okay? And go back to blue. Okay. So there would be a man there. And a bag. And a man. Right. And then, sorry, nag. Nag there. And then if we continue, there'd be a man here, a man here. And then there would be a link there. Okay. And it protects from lysis, okay, bursting. Okay. And there are drugs that interfere with building this, and it makes it easier for the bacteria to be killed. Okay. All right. Moving on. Boom. Do all this up. Okay. All right. So with the gram-positive cell wall, you've got the peptidoglycan, and it's pretty thick. Okay. It also has teichoic and lipoteichoic acid. Hold on. What's this? Hold on. Sorry about that. Okay, so teichoic and lipoteichoic acid help the cell wall to be more stable and to get bigger. It's like a scaffold to build the wall off of. And they also provide a negative charge on the cell surface, okay? And that can be protective for the bacteria as well. If we look here, the gram-negative cell wall, right, is a single sheet of peptidoglycan, 1 to 3 nanometers thick. The thinness gives gram-negative cells more flexibility, but they're more sensitive to lysis. Okay, so it's easier for them to burst. The gram-negative cell wall also allows for the presence of something called endotoxins, in addition to the periplasmic space between the cell wall and the inner membrane. And this is where enzymes that break down toxins can live, okay? And the result of that is that gram-negatives tend to be more often pathogens than gram-positives do, okay, because they have that ability. This is a walk through the gram stain, which is part of what we're going to talk about in lab today, okay? And at the end of the process, what you end up with is gram-positive cells with that thick cell wall, that thick peptidoglycan cell wall, stain purple, while the gram-negatives are going to stain pink, okay? So gram-negative pink, gram-positive purple, okay? And we'll talk about each part of the process in lab in more detail. There are also bacteria that are called acid-fast bacteria, okay? And they have long chain fatty acids in their wall that help them be more disease causing because of their ability to resist certain antibiotics and certain dyes, okay? So you can see here some examples of these different types of fatty acids that you might find in an acid-fast bacteria, right? They come in slightly different arrangements, okay? But you would find a mix of these in a typical acid-fast bacteria. Archaea have cell walls that are made of polysaccharide. Others have cell walls that are pure protein, and none of them have peptidoglycan, okay? So this tells you that archaea, and some don't even have a cell wall, but archaea are much more distant, much more distantly related to bacteria than you would assume, okay? A lot of modifications took place in evolution between archaea and bacteria to achieve that. Some bacteria don't have a cell wall. Mycoplasma don't have this. They are able to resist threats from the outside as a result of sterols that are inserted in the cell membrane. An example is Mycoplasma pneumoniae, which produces something called walking pneumonia. Then there are bacteria that normally have a cell wall but lose it during some part of their life cycle and take on a different shape. And also because of that gap between having and not having a cell wall, they can be resistant to certain antibiotics. And so they cause what are known as persistent infections, okay? Because normally if you have an antibiotic that acts on the cell wall, then you hit the bacteria with it, you hurt the cell wall, then when the bacteria undergoes an osmotic event, it'll burst, it'll explode. But if you've got this period where there's no cell wall, all right, then the drug won't be effective at ruining the cell wall, right? Because the bacteria is not building the cell wall at that time, right? The crew isn't on site. So the drug doesn't work as well. So the gram-negative outer membrane is similar to the plasma membrane. There are lipopolysaccharides here that can serve as receptors. There's endotoxin that can be here. Endotoxin is something that produces disease that is actually attached to the bacteria itself. And porin proteins, which are membrane channels that only allow certain chemicals to get through. Okay, so here we have another barrier to stuff getting into the cell. The cytoplasm has a membrane around it that is a phospholipid bilayer with protein embedded in it. And for the bacteria, this is where ATP gets made. This is where nutrients are brought in. And this is where a lot of biosynthesis happens. This is where, for instance, the cell wall gets produced. It also regulates the transport of nutrients and wastes, and it's selectively permeable. That means only certain stuff gets through the membrane and other stuff is locked out. Just like in the cell membrane of our cells, only certain things can get through, and those would be things that can either pass through a special gate or a tunnel made out of protein, or something that's lipid soluble and can slip through the phospholipid bilayer. Okay. There are differences in the cell envelope between the gram-negative and gram-positive bacteria. Okay, the outer membrane of the gram-negative bacteria provides another layer where antimicrobials can get wiped out. It makes it tougher to kill gram-positive bacteria, more difficult to kill gram-negative than gram-positive. Alcohol-based compounds can dissolve lipids in the outer membrane, and that's one of the reasons that we use alcohol swabs to clean the skin surface before we say do an injection or introduce an IV. All right. The treatment of infections caused by gram-negative requires antibiotics that can cross the outer membrane and get inside the cell. Okay, and those are different from the ones that can present a problem for gram-positive bacteria. Okay. We'll talk about some stuff that may be in the cytoplasm, and we'll talk about sporogenesis. Okay. So the cytoplasm is mostly water, and there's also sugars, amino acids, proteins, salts, ribosomes, cytoskeleton, DNA in there. Okay. It's a pool of building blocks for constructing parts of the cell or for making energy in the form of ATP. The bacteria also typically has a single chromosome, which contains the protein building instructions for the entire cell, right? So that's where the protein building blueprints are, the tools of life. And the DNA is contained in a lump called a nucleoid that is usually attached to a tiny projection. It's a protein that's embedded in the cytoplasmic membrane. It kind of holds the DNA as it dangles from it. Okay. Plasmids are extra chromosomal DNA and can contain genes that provide resistance to certain antibiotics, can cause bacteria to make toxins and certain enzymes. Okay. And these plasmids can travel from one bacteria to another and reproduce themselves. The ribosome is just where the protein is built. Okay. It's what's called a non-membranous organelle. And in bacteria, it's functionally identical, but structurally different from our ribosomes. Okay. Our ribosomes are what are called 80S ribosomes, and the bacterial and the archaeal ribosomes are what are called 70S ribosomes. All you need to know in terms of this is that they are functionally identical, and structurally different from our ribosomes. So what does that mean? That means that we can make drugs that can attack these kinds of ribosomes, right? These 70S, they can go after the 70S, right? But they'll leave the 80S alone. They won't affect them, okay? And that's important because there are some antibiotics that do that. So you can see here an example of what these ribosomes look like, small and large subunits. You don't need to worry about what the S means. The S is just an indication of density, all right? And it's not important. Just know that it means that the ribosomes are different from each other. Just like a house built out of wood is different from a house built out of bricks, okay? Okay, inclusion bodies, all right? They can be used for nutrient storage. They can be used for gas storage so that the bacteria can float. Sometimes salts can be stored here. And sometimes you'll have an outer shell of protein, which can be inside packed of enzymes that work on a specific biochemical pathway. So in this sense, these microcompartments are a little bit like an organelle, a membranous organelle in a eukaryotic cell, okay? The cytoskeleton is what produces shape for the bacterial cell. And there are cytoskeletal components in both bacteria and archaea, okay? The building blocks of the cytoskeletal components are unique to prokaryotic cells and are not likely a good target for antibiotic development, okay? An endospore is the bacterial equivalent of a seed, and it's designed to get the bacteria through difficult environmental conditions, okay? Such as when there's very little moisture or there's the wrong temperature outside, and the bacteria still wants to live. What it can do in some cases is make this little spore, which kind of sits there until the right conditions come by again, okay? Endospores are so resistant that they can handle the vacuum of space, and they can handle extreme pressure and very high temperatures that would normally kill actively growing cells, what we call vegetative, okay? So let's take a look at endospore formation.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
So this is a tough little form of the bacteria that can resist a lot of harsh treatment, okay? Some examples of endospores that cause disease include anthrax spores, Clostridium tetani spores, spores that cause gas gangrene, spores that cause botulism, and spores that generate a condition known as C. diff infection, which is a GI condition that you can acquire in certain hospital environments, okay? We'll look finally at the archaea, okay, they're considered a different cell type, and they're in a separate super kingdom, and they're actually thought to be more closely related to us than they are bacteria. And that reason for that is their DNA similarity to ours and their ribosomal similarity to ours. Okay. Now, the thing about archaea is that they have some unique properties, right? They have a novel method of compressing their DNA. They have unique membrane lipids and cell wall components. But the main thing about them is they really like extreme environments, such as very cold, very hot, very acidic, very basic. You name an extreme environment, you're going to find an archaea that lives there. Okay. Some live on the human body and might be capable of causing disease, but none that really we've described yet. Okay. So most of the disease causing organisms that we're going to talk about in this course are going to be either bacterial or they're going to be eukaryotic. Okay. But this just shows you some of the different characteristics that you can look at between the different cells. Okay. We're going to talk a little bit about how we class bacteria in different species. And we're going to talk about older versus more modern methods. Okay. So there's a book that's both online and in hard copy called Bergey's Manual of Systemic Bacteriology. And it's a list of how closely related different bacteria are to each other. And this information came from genetic information. Okay, so what they did was, in this case, they sequenced the genes for the ribosomal RNA that's used in these bacteria, and they compared how similar they were. And the more similar they are, the more closely related they're going to be. The older method is called determinative bacteriology. And here, what we do is we look at the bacterial shape, whether or not they have flagella or pili, or the arrangement of the flagella or the pili, the way they stain, and the way they grow on different kinds of media. Okay. So this is now the old fashioned way of doing it. And it's sort of the way we're going to do it in this lab. Okay. And these techniques are still useful enough to provide information about what kind of antibiotic to select for people that are infected with these different types of bacteria. The different divisions of bacteria and archaea include gracilicutes, which are gram-negative bacteria with thin cell walls, firmicutes, which are gram-positives with thick cell walls, tenericutes, which are bacteria that don't have a cell wall and are capable of different shapes, mendocicutes, which are archaea. These are primitive cells with unusual cell walls and different modes of getting nutrition. Bacterial species are a collection of cells, all of which share similar pattern of traits and between 70 to 80% of their genes. Okay. A subspecies is a bacteria of the same species that has different characteristics, and a serotype is a representative of the species that has different interactions with certain antibodies. Okay. So what we're getting down to with subspecies, strain, or type and serotype is a smaller and smaller group of organisms as we continue from genus to species to subspecies all the way to serotype. Okay. Now, again, you want to ask the big question, which is, why do we care? Okay. We care because this allows us to pick the right drug to kill pathogen, the bacteria or the microbe that causes the disease. Okay. So all of these things seem, if you just look at them separately, they seem kind of, you might look at that and say, well, whatever. But it really isn't whatever. Okay. If you know certain characteristics, then you know what drug to pick to wipe out the infection, right? And that's going to be a better outcome for your patient. Okay. So that's why what you do in microbiology is you play detective, right? You figure out, what are my clues, right? Does it have a cell wall? How does it stain? What does it grow on? Okay. What do the cells look like under a microscope? And then I can say, okay, it's this kind of microbe, so I should select this type of antibiotic to take care of your infection, right? So that's the whole idea behind learning about this stuff. Okay, that's why we talk about it. All right, we'll break here. I know I went a little over, and in lab we'll hit the chapter 4. Okay, and I'm going to send you the notes in between now and when we come to lab, and then we're going to talk about gram staining today in lab, and I will send you guys a lab to do just like I did last week. Okay, so before we bump out, are there any questions? We're good. Okay, I will see you guys in lab. Okay, well, I will see everybody in lab. All right, and I will send you that material.
Okay.